Okay, I think that we can start. So hello, everyone. This is our 10th colloquium. So time really flies, and hopefully we'll have a lot more talks also next semester. So in these colloquiums, as many of you know, we've had numerous discussions about the solid-liquid interfaces, things like molecular interactions, absorption, and many other things that constitute electrochemical phenomena. So recently, I would say probably in some way it prompts the questions that one can ask: can we really use these electrochemical? Can we really see these electrochemical processes directly at the nanoscale using a microscope? And what kind of limitations and difficulties will we really face if we try it? So one way is to use electron microscopy, as it's one of the few microscopy techniques that can provide an atomic resolution. So there has been some hope for a while that someday electron microscopy may help us to get a real glimpse at what's happening in the electrode, and especially at the solid-liquid interface during all these electrochemical reactions. But once you start planning these in situ experiments inside an electron microscope with real liquid and under applied potential, you will quickly realize that there are millions of challenges that make the whole task almost, I would say, insurmountable. So another thing that you will probably realize is that your knowledge and experience with conventional electron microscopy, which is done under high vacuum, is hardly helpful. And normally, what we do in such cases is that we break down the big problem into small pieces and then address those pieces one by one, hoping that maybe at the end, we will be able to arrive to a point when we get closer to solving this big or grand challenge. So today we're hosting Professor Francis Ross from MIT, who, in my opinion, fearlessly took on this considerable challenge and has addressed many issues that are inherent to in situ liquid cell electron microscopy. So Frances received her PhD at the University of Cambridge. She spent almost 5 years as a staff scientist in NCEM, which is the National Center for Electron Microscopy at Berkeley. And from 1997 to 2018, she had worked at IBM. And then since 2018, she's a professor in the Department of Material Science and Engineering at MIT. So with that, Francis, please, now the stage is yours.
All right, well, thanks for the intro, Andrew. I'm going to go straight to the screen share and then start talking about all the things I'd like to talk about today. So I really appreciate the opportunity to present this work to an audience of real electrochemists, and it will probably become obvious that I'm not much of an electrochemist. I'm a microscopist primarily, and what I want to do is try and bridge the gap between the two communities. Microscopists tend to see everything as a microscopy problem that can be solved by microscopy. Electrochemists are a little bit suspicious of all of this, and so what we want to do is see if there's a good way to kind of make the join. So I'm going to talk about how you can see electrochemistry in action using liquid cell electron microscopy. And before I go any further, I want to acknowledge all the people. So I'm going to show a lot of different things centered around the work that we've done in our group, and with some references to work that's going on elsewhere. So the people I'm most acknowledging are my current students at MIT, students and postdocs, and especially the key contributions at IBM. So this was all developed at IBM. There were some technical challenges to be overcome in the early stage, some motivational challenges, so we got through it. But this is really down to the work of Mark Reuter and Art Ellis, who are technical experts in building things and playing with vacuums, and Rutramp, who really is the intellectual driver of all of this. So with all these other people that we've worked with over the years, it's been a really fun and exciting journey, and I'd like to thank all of them and also the funding agencies who have supported the work. All right, so let me talk firstly about why should electrochemists even bother with transmission electron microscopy, which, as Andrew said, is primarily a technique where you put a material in a vacuum system and you fire electrons through it. And so this is not really a friendly situation for doing electrochemistry with liquid phase components to the experiments. But there's a huge motivation, and the motivation is that direct observation is really important for electrochemistry. A lot of the models that have been developed are developed based on ensemble measurements. You know, what is the final cluster density in nuclei per square nanometer? What's the average film thickness? What's the average current density in the system? But fundamentally, these growth models are really applying them applicable to individual structures like nuclei pits or dendrites. And so we need a way of linking the individual objects to the ensemble measurements that create them. So there's a lot of ways of doing this kind of microscopy. Light microscopy is a fabulous way of getting good images of things as they change, high time resolution. Of course, the spatial resolution isn't so great. Scanning probe microscopies can give you excellent spatial resolution, but typically, because the probe physically has to scan, it takes time. I know there's some fast ways of doing it, but generally it's a slower process, especially for rougher surfaces. And so electron microscopy can occupy a kind of a niche where you can record images with a field of view of ranging from tens of nanometers to microns. You can record images at hundreds of frames a second, assuming the sample can stand the dose. And so in between these other microscopies, electron microscopy has a real role to play in bridging length scales and furthermore, linking movies of things happening or images of things happening with simultaneous measurement of electrochemical parameters. All right, so that's why we got excited about this. Of course, being in IBM, the primary driver was the electrochemical production of copper interconnects for microelectronics. And so that led us down a pathway that I think has been a whole lot of fun all along the way, as well as incredibly frustrating at times. So here's what I want to talk about in this presentation. I was instructed to break it up into two parts so we can all take a breather in the middle. I'm going to firstly give you a little bit of a historical perspective on how this all developed and what's going on. I'll talk about the fundamental kind of engineering aspects of how do you design a way of looking at liquids in the microscope. And I'll talk a little bit about resolution and dose. And then we'll talk about some examples. I'm certainly not going to review the field, but I want to give you a sense of the scope of things that you can look at with liquid cell electron microscopy in terms of basic physics of crystal growth, corrosion processes, battery reactions, and some related topics. And then we'll take a break, and I'll be happy to answer questions about all of this, because in the second part, I want to go on and talk about the challenges and what we can do to improve the quantitative nature of this technique, and then what the opportunities are and the prospects for future advances using this technique. All right, so the motivation for this talk, I'm reproducing here a direct quote from the email that Andrew sent me when he invited me to give this presentation in the first place. So you know this technique exists, you're kind of intrigued by it, but you think it's going to be dreadful, awful, take too long, not give quantitative results. So by the end of the talk, I hope that I can answer these questions here. How do you do this in the first place? Why is it worthwhile? Is it difficult? Well, yes, but it can be done. Can I believe the results? And can it ever be really quantitative such that it contributes real understanding to electrochemical processes? So thank you, Andrew, for the invitation and for this dramatic statement in your email, which I think has really inspired me to look closely at, where this is going to go and what we can do to improve it. So I want to start by setting a little bit of context, since I know that you're primarily electrochemists rather than electron microscopists. But electron microscopy has had a kind of a fantastic growth spurt over the last few years. Almost every aspect of electron microscopy has seen dramatic improvements, not just incremental things. We could look at the improvements in detectors, where we can count every electron that goes through the sample and we can get high-speed imaging. We can consider aberration correction, which improves the ability of microscopes not just to give high-resolution data, but to give more interpretable data and to give good images even at low kV. We can also look at things like image analysis software. You can imagine if you record 400 frames a second for a few minutes, you have a lot of data there. And so you need ways to kind of pick the stuff out, the good stuff out of it. So there's a lot of development in these areas. So conventional microscopy is in a fantastic state, but we can also look at improvements in situ microscopy, the idea that you do something to the sample while you're imaging it and record the changes to understand reaction mechanisms. And so in the field of liquid cell microscopy, I think it's fair to say that ever since the invention of the electron microscope, it's been a goal to image liquid specimens as easily as you can do so in a light microscope and to measure liquid processes with good spatial and temporal resolution. And because of the advances in sample preparation and many other things, we've seen a lot of progress on this problem in recent years. And I'm talking about not just electrochemistry, but for example, imaging biological materials in liquid rather than using cryomicroscopy. And I'm not just talking about static images, but recording movies of things that take place in the liquid phase, such that you can really record the kinetics of processes as they take place. Okay, so how do we do liquids in the microscope? As you know, the microscope is kept under a pretty good vacuum because you want the imaging electrons not to hit anything else apart from the sample on their way down the column through the lenses and onto the detector. So normally the microscope is held at a high vacuum. So you can imagine, but there's no intrinsic reason not to put a liquid sample in there. So there's no problem with having a liquid in the TEM if it has a low vapor pressure and it won't boil away. And so many catalysts are liquid phase. This is gold germanium eutectic droplets that catalyze a semiconductor growth process. They're completely liquid. They just look amorphous in the microscope, but they're fine. They have a really low vapor pressure. You can also, of course, look at liquids enclosed in a solid matrix. So here's a liquid lead, solid lead. You can look at these phase transformations. The lead is hidden away. It's trapped within a solid, no problem at all. So this is great, except, excuse me, water is the most exciting liquid and it has a very high vapor pressure. So we have two ways to approach this. And I want to emphasize the dates on this slide. Look at this, 1940s. The microscope was barely invented at the time. The concepts, the engineering were being done for electron microscopy, and already the pioneers were saying, we don't just want to look at solid metal foils or whatever, we want to look at liquids. How can we do this? And they had already developed the two techniques that really are the ones that are in use today. So on one of them, we have differential pumping. The sample is in one part of the microscope column. The pressure there is quite high because it's got water and it's it's evaporating. It needs to be at equilibrium in a atmosphere of water vapor, and so you have differential pumping with small apertures and pumps on the other side that make sure that the rest of the microscope is under vacuum. So the electrons pass through the column, they pass through some region of poor vacuum, and then the rest of it's okay, and that's fine. You can get decent images. So this is a technique now called open cell electron microscopy. The other way, window cells, proposed in the 40s again, you put your liquid in between two windows. And these were a few microns apart. The water condenses. The trouble they had was, what would we make the windows out of? Without modern materials techniques, they were made of kind of uncontrolled thick materials. They weren't very good. And so what happened at this time was that the resolution was poor, not much better than using a light microscope. And the environment was an uncontrolled mix of water and air. So the biologists turned away from this and they decided to develop cryomicroscopy, and the material scientists found other problems to address until modern materials and fabrication techniques changed the game for us material scientists. So this is a SEM picture of a typical window chip. It's a silicon nitride window on silicon. And the way you make these chips is that you start with a silicon wafer. You deposit silicon nitride, typically on both the top and the bottom surface of the wafer, and then you pattern some holes on the bottom surface, you etch away the silicon nitride, and then you etch through the silicon to the top surface, which is underneath in this image, and you control the etching such that the window, the silicon nitride, is preserved. So here's a cross-section of how you would make two of these chips into a closed cell by putting them face to face with a spacer layer between them. And the magic of silicon nitride is that you can develop it. It's amorphous, doesn't cause any contrast in the microscope. It can be very thin, a few tens of nanometers, and yet incredibly robust. It can stand up to it can stand atmosphere pressure differences across it without breaking. It's under tension, so it's kind of stretched flat, and it's just fabulous stuff. Graphene is another choice. It's thinner, that improves the resolution. We'll discuss that later, but it's a bit harder to control in a manufacturable way. So silicon nitride is the most popular material for liquid cell microscopy. So this is the closed cell version, and then the open cell version, where we're using low vapor pressure liquids that don't even need to be enclosed in a box. We can use modern techniques that, for example, with piezo-driven components of stages, to make electrical contacts and develop open cell microscopy. All right, so we have our two ways of imaging liquids in the microscope. And I'm going to mention the open cell work, but I'm primarily going to talk about the closed cell work because I feel it's a bit more flexible and realistic for doing electrochemistry. And so I'm going to focus more on how we build these closed cells and what we can use them for. So here's a cross-section of an early closed cell. They're not done like this much in these days, but this gives a good example of what you can do. So we have our bottom window. We want to put liquid in a thin layer and have the top chip on top. This is all the top chip. It's just there's a cross section, so there's an opening in the top chip. You can inject the liquid down the hole through the gap by capillary action, and then out the other side. You seal up the whole thing, and then you stick it in the microscope. And so we want to do electrochemistry. We have to put some electrodes, and we put them here on the bottom chip. They're made of stripes of gold in this case, or platinum or something, and they extend over the electron transparent window. So the path of the electrons is through the vacuum of the microscope, through the top window, through some layer of electrolyte, that's usually hundreds of nanometers, through an electrode, which has to be electron transparent, through the bottom window, and then back out to the microscope vacuum, and then to the projector lenses and the camera. All right. So if you took an optical microscope image of this sample after it had been assembled, we would just be looking at this. The important thing here is the imaging window. It's typically 100 microns, sorry, that says millimeters, it should say microns, 100 microns square, let's say, or a strip, or whatever shape you want. It's got silicon nitride covering the whole thing. You can see the two electrodes. And importantly, the region that you look at in the TEM is much smaller than the total window. But it's a bit hard to get the windows really small you have trouble aligning the top one with the bottom one. So normally we have a small field of view in the electron microscope and a much larger field of view if we want to do some optical measurements. So we can see processes going on the electrode in plan view, or we can see things going sideways around or off the electrode, or we can see processes at the perimeter of the electrode. Right now, the good thing about this is that you run these electrodes to an outside connection. You can have basically as many as you want, and you run it through a potentiostat. So we have a simultaneous recording of the electrochemical conditions and the movie. Okay, so the liquid cell technology has evolved greatly, I would say. And in the early days, this was all done by hand. Everything was just glued together by hand. You can imagine the success rate was pretty poor. So, so Mark, who was our first student to work on this, he got about 1 in 20 that worked. And that was a bit grim, but he plowed through it and he got fabulous data out of it in the end. This was the early liquid cell that he worked on. The pieces were attached by glue. There was a kind of a reservoir to put the liquid in, and then we could put the electrodes in through that, and they were wires. And then the working electrode patterned onto the substrate and then connected through the silicon to the outside world. So you can obviously make these in many different ways. You can do wafer bonding, and you can do more sophisticated versions where the two chips are not glued together but are squeezed by O-rings. And this provides a much easier way of assembling the chips and getting the spacing that you want and the configuration that you want. And it's possible, of course, to pattern quite sophisticated electrode designs onto the chips so that you can at least make an effort to replicate real-life electrochemistry, for example, in these, I mean, with a flat geometry. Remember, the liquid is quite thin, but this is like a Healy-Shaw cell. Okay, so now you can just go out and buy this stuff. You don't have to do it yourself. There are 3 major companies, a number of smaller ones. I'm not going to kind of talk about the benefits of each one. I just want to point out that they all have very good web pages. You can look this stuff up. You can, all you need is money, basically, and then you can buy a complete setup that should fit in any microscope that you have. You can see their integrated systems with a sample holder. The chips are placed on the end of the holder. The liquid flows through little tubes down the length of the holder to the chip and back out again. So you can refill the liquid or change the chemistry as you go along. You can do all the functions of the microscope: analytical, microscopy, high-res, STEM or TEM modes. All the different things that the microscopist would normally be able to do can be done excuse me, using these sample holders. Okay, so before I show you some examples of what we can do with this equipment, I want to make a couple of comments that are a bit sort of basic, but we'll go into more depth later on. And the first comment is about resolution. It might, excuse me, it might seem a bit surprising that you can get high resolution using these kinds of samples, but you really can, especially if the liquid is thin, if the windows are thin, if the windows are made not of silicon nitride, but of graphene, which is really thin, not single sheet, but multi-layer graphene. You can see at the atomic level, the structures of objects within the liquid. So these are not electrochemical examples I'm showing. These are simply other, these are other crystal growth, phase transformation kind of experiments that people have done where the atomic resolution is visible in the images. So high resolution is possible. Even in biological materials, you can see these viruses, there's some structure in here. You can see the motion of particles, you can see the sintering of particles as they approach and attach to each other. And you can see all of this with high resolution. So high resolution is possible, but if we're doing electrochemistry, we have actually a harder job, because we don't simply want to optimize our sample for high resolution. We want to optimize it for understanding physics. And that means that the things that give high resolution, which is very thin windows without any kind of functionality to them, or very thin liquid layers, those things don't work so well for electrochemistry because thin liquids don't give realistic environment for doing our electrochemical reaction. For example, if you're looking at diffusion limited nucleation and growth, you need a realistic diffusion field. You need some volume around your object as it grows to supply the ions. You need a pattern of electrodes, and the electrodes have to be robust and they have to be electrically connected. So we're not able to choose the sample only for high resolution. And we're going to talk about the trade-offs a bit later on. But the main picture is that atomic resolution is possible in liquids, but not always achievable, or you don't always want to achieve it in electrochemistry if something else is more important to you, like setting up a realistic diffusion field. The other thing is electron beam effects. We will talk about this a lot later on. Water responds very strongly to the electron beam in a way that's kind of different from how solid materials respond to the beam. I think electron microscopists know that every time you put your sample in the microscope, you're exposing it to quite an inimical environment. But for water, you really see the effects. You get radiolysis, you can form things like hydrogen, you can change the chemistry by creating hydrated or solvated electrons. You can use those effects to reduce metal ions, and you can scan the beam even to make, these are localized within the beam, and you can even do some little writing exercises. So I put this one in just for fun because you can see we were operating this by hand. It's not really programmed. As we scan the beam around, the changes in chemistry caused by the beam result in precipitation of individual gold atoms from gold ions in solution, and you can use it to write the names of your institutions. That's what people tend to do. And so we reckoned these were nano letters, well, micro letters, and we ended up submitting this to the journal Nano Letters. So that was kind of fun. Anyway, so I'll talk about this a bit more later on, but you can get bubbles, changes in pH, you can change the chemistry of the solution. You have to be on the lookout for artifacts. So that was a quick survey of the history and the current status and the equipment for liquid cell electron microscopy. So now I'm going to talk about what you do with it once you've set it up. And I'm going to summarize some experiments in nucleation and growth, that including growth fund propagation to test and refine electrochemical models. And I'm going to talk about corrosion and galvanic displacement and some other things that can be done, batteries. Okay, so the first thing I want to show you is something that I found especially interesting at the time. It was one of the motivations for doing electrochemistry was, as I mentioned, how does copper grow when you're trying to make an interconnect for a microelectronic circuit? So here's an experiment where we're looking down onto a gold electrode. You can't really see the structure of the gold electrode. It's just this kind of fuzzy gray background. But we're in copper sulfate, sulfuric acid solution, and we're applying a constant potential with respect to a copper reference electrode. And we're going to look at what happens. And so this is one of these is the movie I showed at the start. You can see nucleation and growth of individual copper islands, while you simultaneously have the current time graph. So the current transient has the behavior we expect. Also, it has a peak and then a decay. If I apply a higher more negative potential, I should say, you get a higher density of smaller objects, and the reaction takes place more quickly within a few seconds. And then if we go to a massive 100 millivolts, you can see a really high density of small objects, and the whole reaction's pretty much over in a few seconds. And so this would be hard to do. You can imagine in a scanning probe, instrument where the reactions take place over such a short time scale. Now, to illustrate how quantitative this can be, we can pull out all of these current time transients, and we can compare them with electrical models that predict that nucleation and diffusion limited growth should go through several stages, right? You have the nucleation of the objects. They could be progressive or instantaneous nucleation. There's some early stage growth going on. Diffusion fields develop, the growth slows down, and eventually at later stage growth, all the diffusion fields have overlapped and the growth rate of each object decreases even more. So this is what we expect from models. How does, and indeed these graphs show what they ought to show in the models, but how does that compare with data? We can get a lot of information out of these videos. Now, each frame is a pretty bad image. There's not, you know, it's not a beautiful image, but you can track the evolution of these particles in a very, you know, accurate way using image analysis. And you can, for example, measure the nucleation kinetics from the number of clusters per unit time. You can see the shapes and the growth rates of individual clusters, and you can work out correlations between island positions. And the quality of this data really surprised me at the time, because you can see this is one individual particle. This is its radius versus time. On a log-log plot, you can see that the growth rate has a change, that it goes from growing quite quickly to growing quite slowly, exactly as predicted from the nucleation and diffusion limited growth models. And so the models work to a great extent, but there's a big discrepancy between the parameters you obtain, for example, for nucleation density, from fitting the current time transients, these ones here, and what we actually see in the movie. And so the way that this could be resolved in, you know, in understanding what's going on is to say, we think that the simple model of the copper ions arriving from solution onto the existing nuclei is not complete. We have to consider two diffusion processes. They have to arrive on the electrode, and then they have to diffuse laterally to arrive at a nucleus, and those processes have different diffusion parameters, and therefore you can expect different kinetics compared to a model that imposes a single diffusion parameter. Okay. All right, so you can go further with these kinds of deposition experiments, and I want to show you another sort of ugly video. Do you see on this one, sort of a, you know, again, the resolution is not great, but you can see very clearly the shape of the objects is quite different from the ones I showed earlier. And here we're growing copper in an electrolyte that has some of the commercial additives that are used to control the growth when forming interconnects, and it's possible to evaluate the process of the, you know, the effect of each one of these things by observing in the microscope how it changes the growth morphology. All right, here's another example. We've grown copper on the electrode, and now we're going to grow so much copper that it starts growing sideways off the electrode, and we can see how the growth proceeds as a function of time. Again, a very short video, I did speed it up a little bit, but what you can see in the video is that the growth starts relatively flat, and then it becomes rougher and rougher until we get these dendrites formed after a few seconds. And so the high deposition rate leads to a growth instability. If we grow more slowly, the movie is actually much less interesting, because really not a whole lot happens. You can see it's growing. You can see this, and now the movie is just repeating itself. You can see just the growth of the copper front, and you can see that there isn't much in the way of development of dendritic structures here. Okay, so what can we do with these things? We can compare growth under different conditions. We can look at the height of the peaks and the valleys. We can look at the roughness and how that evolves with time. And we can compare with kinetic roughening theory or diffusion limited growth. We can evaluate what the exponents for this roughness versus time log-log plots are expected to be, and then we can match this with the behavior that we see under different conditions. And what we find is that, again, as another diffusion process comes into play, the transition time between these two regimes is not what you'd expect, and lateral diffusion is an important factor here. But maybe the most kind of universal thing here is that these tall instabilities, they grew from small bumps formed by kinetic roughening. So however much you polish the electrode surface, it's not really going to help. The surface intrinsically will become rough, and then intrinsically will form dendrites. So we have to suppress instabilities through means other than the initial surface flatness. And one of them, a well-known way, is to use additives. And so here again, liquid cell microscopy can evaluate the effect of additives, such as in this case, bismuth, on the nucleation and growth of zinc, to show that you can get a slower evolution of roughness if you have the additives in there. And then I think the last one I'll show you for these is kind of a fun one, because there's a lot of charging regimes where the current and voltage change quite dramatically with time. And so pulse plating is one of these very important processes where we stop depositing for a few moments to allow the diffusion fields to catch up. So you can track that in the TEM. But pulse reverse plating is the theory whereby you add some material and then you reverse the voltage. It knocks off the asperities, and then when you continue to add more material, maybe it grows flatter. So I want to show you how this works in the microscope. And you can see a pulse and a reverse on each cycle. But instead of making it smooth, this actually seems to lead to a coarsening effect. So this is one of my favorite movies. I don't know scientifically how much we can learn from this, but just visually to show that this sort of inequality gets exacerbated during growth, because the larger object is just stealing all of the material that's being released by the smaller ones when you do the etching. Okay. All right, so I talked a lot about nucleation and growth and matching with growth models. I want to briefly cover a few other things that liquid cell microscopy has been used for. One of the most important things is corrosion. There's a number of groups studying this. These images are from one of our experiments evaluating the corrosion of aluminium films in salt water. These are slow experiments, but you can see the pitting corrosion followed by the formation of tracks beneath the oxide. And you can see intragranular pitting in some of these experiments also. We can also do electrochemically driven corrosion, whereby we're recording we're slowly scanning the potential and imaging the film as it dissolves. And we can get reasonable electrochemical traces from these experiments to correlate with the results. And other groups have done, for example, corrosion around particles in stainless steels and similar types of experiments. Here's a corrosion experiment where we're etching away the one component of a bimetallic particle. So in this movie, you can see things that were originally kind of solid polyhedra, and they end up as these little finger-like structures. And what's happening is that the initial structure has platinum deposited as these kinds of lines. And then as you etch away, and the nickel fills in the space, as you etch away the nickel, you end up seeing the platinum structure. So the idea here is to control the surface chemistry by rebalancing the nickel and platinum in these catalyst particles. Okay, and finally, galvanic displacement. I think finally, this is an experiment where we don't apply a voltage, but we're flowing palladium ions over a nickel electrode, and you can see the formation of solid palladium and the dissolution of the nickel. It's not all electrochemical, there's electron beam effects here, but the principle is in there. Okay, so the last topic I want to cover is maybe one of the most important ones. What happens inside a battery during operation? And this has been addressed by a number of groups, and there's some really innovative and insightful work out there. I've just picked a few examples. For each of these, what you see clearly is the correlation of images with electrochemical signatures. So we can track how things evolve, both by looking at the electrochemistry of the whole chip and by looking at the behavior of the individual objects that make up the reaction. So we can look at the formation of the SEI, we can look at the formation of dendrites, we can measure the composition of these things, we can look at the intercalation or the reaction of lithium with different materials through energy, compositionally sensitive imaging. We'll talk about this a bit later on. So there's a lot of work going on in this field, and some of it is really, really good. I'll finish with one movie to show how open cell microscopy works. I mentioned this earlier as a technique where if you're lucky enough to have an electrolyte that's not very, doesn't have water in it and isn't going to evaporate, you can actually look at it in the microscope without windows. You just need to make the electrical connection. So these experiments, these are some of the earliest experiments with the open cell showing lithiation of nanowires, where the lithium is brought on in an ionic liquid electrolyte along the surface of the wire to do the reaction. Okay, so this is where we are now. The story so far. I hope I've convinced you that you can visualize a lot of different phenomena, both general liquid phase and electrochemical processes. This technique is used by quite a number of groups. There's many publications, review articles, there's dedicated symposia at conferences, there's even dedicated conferences, and I'll advertise one right at the end of all this. You can buy the equipment and you can train students, and you can hire students who have been trained by somebody else. So this is the happy scene that I want to leave us with after the first part of my talk. And after the talk, we're going to go on and look at some of the sort of darker side of it and discuss what the issues are, as well as the opportunities for the future. So I'll stop sharing now and we can take a break, and I'm happy to answer questions.
Thank you, Frances. I think in principle, you can keep the screen on. That's okay, because probably people will be asking about the slides too. Just
okay. Well, if somebody wants it, we can.
The audience is unmuted. Please feel free to ask questions. You can raise your hand or you can just directly ask. Okay, we have one. Go ahead, Rana.
Hello, very interesting talk. I really enjoyed. So thank you so much for sharing insightful information. So I have one question. I'm not very expert, but my question is related to like in situ TM, microscopy or even a liquid cell. So we reduce the volume too small, like it's in some microliters sometime, right? But when we do reaction, it's like in sometime milliliters and sometimes liters when we do nanoparticle synthesis or catalysis synthesis. The problem of aggregation, agglomeration, and growth, they are like very different dynamics maybe in the big flask and as compared to the in situ or liquid cell TM. So how we can assure that both processes are correlated? I mean, what we are observing in the liquid cell, it's translatable to the volume in the flask.
That's a really good question, and I think is one of the key things that you have to think about when you choose your method of analyzing any problem, right? So if you are looking at, so the first thing I would say is that for a normal, for any electrochemical process, you have to think about the effect of having the small volume available, and especially the small area electrode. And so as an example, you may do a diffusion calculation to work out how do the diffusion fields develop around my electrode, given that I know exactly how many ions I converted. If I'm growing copper, for example, we can work out how many copper atoms have been placed on the electrode. Where did they come from? What's the diffusion field within the geometry of the liquid cell? And so if that is completely, so often if you do a slower reaction, it's fine. Things can move in from a large distance. You expect to see similar behavior to a bulk system. But in cases where the diffusion, let's say the environment is different, you may want to rethink which technique you're going to use. So can you, for example, use light microscopy? Can you label, can you get some signal out of the particles that would be visible in a larger volume, maybe X-ray microscopy? And then can you compare it with the TEM and say, we know it has this limitation, but we think that a correlation between different microscopies with different spatial and time resolutions and different volumes would be valuable. So I think that there's no one answer to your question, except to say that you should think about this when you apply the technique and say, is this the right technique for me? Or is the diffusion limitation going to be actually too difficult to handle?
Great. Thank you so much for the answer.
Hello, it's Ismail. May I have a question?
Yeah.
Great talk first. Thank you. And just have a question about the thickness of the liquid that actually, or how would be the electrolyte? I mean, in terms of because I mean, if it's not kind of uniform, and then we have a lot of bubbling issue. I mean, as soon as we go to some very low or very high potential for reduction or oxidation potential, right. I mean, there is like different ways that we can use, like the syringe pump or peristaltic pump, or I mean, different technical methods that we can kind of solve this problem. But in your point of view, which one would be the best?
I think
to just get rid of this kind of bubbling issue and then have a uniform thickness of the liquid.
So it's a really important point you raised, that if you want to understand the details of the electrochemistry, you have to know the geometry of the liquid.
Exactly.
If the electrolyte is not in a uniform, I mean, it's bad enough that it's thin. So it's maybe it's a few hundred nanometers thick, or maybe it's only tens of nanometers thick. But at least if it's uniform, you could calculate the diffusion fields. If you have a lot of bubbles, you have the danger of actually losing connection in your circuit, because the bubbles in a liquid cell can't get away as easily as they can in real life. So I think that the strategies for this depend a little bit on the system you want to study. So you can always say, is this the right technique? Is the bubbling going to really, should I be studying something that creates a whole load of bubbles using liquid cell? Could you maybe use deliberately a much thicker liquid, because the bubbles are highly visible if that's the subject you want to study? Or could you engineer the contents? Bubbles always go to the thickest regions. Can you engineer the cell such that the bubbles are drawn away and pulled out of the field of view into a larger reservoir? And that's a possible thing. So if the bubbles are the main object of the study, then you can't look at them when they get too large. The TEM is really only good for the smallest of the, just after nucleation.
Yeah, because some people I've seen that they just kind of use the static mode, like without flowing the electrolyte. And how do you comment on this method? I mean, if we just stop everything and then just do the Imaging.
Right. No, actually, I want to comment on that a bit later on. I'll show a slide about that. That if you think that flowing the liquid will kind of carry away all the reaction products, you'd have to flow it pretty quickly for it to have any kind of benefit. So I think that flowing the electrolyte is a beneficial thing in terms of making sure that the environment is somewhat constant and that there's a uniform liquid layer. But in terms of actually using that to fix things like bubble formation, it has somewhat limited benefits. So,
Thank you.
So yeah, okay, right.
Thank you, Professor, for a very interesting presentation. My question is, can this technique be adopted for high temperature systems? For example, molten slags.
Yes, good question. I think there are two aspects to this. So your high temperature is probably a bit different from my version of high temperature. I'm going to show later on that you can heat the sample. You can put small heaters in liquid cell chips, but we're talking about temperatures of less than 100 degrees C. If you want to study very high temperatures, you run into a problem, and this is true not for liquid cell, but in general for electron microscopy. If the temperature is very high, the atoms vibrate a lot, and the contrast in that they provide, in terms of imaging with electrons gets lower. So there's something of a limitation when you go up to hundreds or 1,000 degrees. You can record images with samples. People have looked at the sintering of ceramics at 1,400 degrees C in the microscope, but you don't get really good images because you get a lot of thermal diffuse scattering. So for liquid cell microscopy, I would say the technique hasn't developed beyond 100 degrees C, but in principle, you could locally get high temperatures. You just can't put a lot of heat energy in. If you can reduce the volume that gets heated, then you can, in principle, heat it up more. And you may have to find high temperature materials to construct the liquid cell. I think that's like an engineering problem rather than a fundamental problem. So basically you're asking a difficult question, but I think that it may be partly answered by engineering innovations. Right.
Thank you for the very, very useful input. Thank you.
Okay, great.
Thank you, Francis, for your nice presentation. Just I have a question. I'm not working in this area, but it's very interesting for me. Because for TM imaging, we use a gun and it's produce high density of electrons for imaging. Mm-hmm. And then this electron, how doesn't participate in the electrochemical reaction or doesn't make any interference during this reaction?
Absolutely makes an interference. So the electron flux, you're asking, these are all great questions because we're going to discuss in the next part about what happens when the electrons hit the water. And they don't, it's not necessarily the case that they go into the electrochemical system as like an additional current. You typically don't get high energy electrons absorbed by the sample, but what they do is change the chemistry of the electrolyte. So they, for example, would change the pH of the electrolyte by doing radiolysis reactions with the water. And so the net result is that the electron beam can affect the electrochemical reaction quite a lot, but not through kind of adding electrons into the circuit or doing the job that the electrochemical potential is supposed to, is doing at the time, if that makes sense. They have kind of an indirect reaction through the electrolyte.
So, for example, they don't participate in electrochemical reduction or something. So
they don't directly participate in electrochemical reduction, but if you put high energy electrons through water, they create hydrated or solvated electrons from the water, and those are perfectly able to do reduction.
Okay.
Of ions in solution. So all of the effects are like one step indirect. It's not the direct primary beam, it's what they do to the electrolyte that causes changes to the electrochemistry that you're looking at.
Thank you, Francis. Yeah. Thank you, Andrew.
I'll show you that in a few slides, yeah.
Francis, there's one question from the chat that it's up to you if you want to leave it for the final part. So it's about the spectroscopy in situ. Do you want to comment on that now? Or,
yeah, I'll comment on that. Yeah, so spectroscopy absolutely can be used in liquid cell microscopy, and you have to worry about two things. One is sort of the logistics, the geometry of the sample. So as you can imagine, in a normal sample, it's kind of flat, and you shine on the electrons, maybe you collect up the X-rays. If you try and do this with a liquid cell, you have to get in and out of the thin region. And so you can have some geometrical issues, because remember, it's down the bottom of a hole in a silicon wafer. So if you solve those problems, which has been done, then you can do EDX in the microscope. And similarly, energy loss spectroscopy, which uses the transmitted electrons, that's completely fine as well. But the trouble with both of these spectroscopic techniques is that they typically need a much higher dose to get a good piece of information. I mean, you know, the cross section for inelastic scattering is low, and so you have to put a lot of electrons through the sample in order to get the signal out. So what you end up with is you may make a measurement, but you may also destroy the sample in terms of, you know, beam-induced effects while you're trying to do your experiment. And so one of the examples I showed did involve some energy loss spectroscopy. I'll show another one later on, but spectroscopy is possible, but advisable really if the sample is kind of robust enough to stand up to the electrons.
Great, thanks. Francis, maybe a somewhat relevant question that I will ask, the last one before we continue. So at the beginning, you showed really some nice historical pictures of people assembling an electron microscope specifically for the liquid cell microscopy, right? 1940s or 60s.
Yeah.
So today we're all in some way locked to the existing manufacturers in terms of the column, the design of pole pieces, and so on.
Right.
So if there could be some type of customization or improvement that we could make to something up to the microscope and not to the holder itself, what would it be? Would you say that, okay, we're limited really by the amount of space that you have for the chips, or we're limited by whatever inability to bring liquids right in, and we have to flow through the holder, which might be not an ideal situation?
Yeah, that's an excellent question. I think that the one thing that if you were designing a microscope just for electrochemical liquid cell experiments, and let's say the closed cell version, right, the window version that we've mostly been talking about, the one thing that you really want is a local calibration, quantification, access to the sample by other probes, like, for example, light, or even X-ray microscopy, or having a temperature measurement locally near the sample, or having say by infrared detection, or for example having as you mentioned, having a better way of delivering the liquid, whereby you have a much larger kind of way in until you get really close to the sample. You can't do much about the fact that the liquid still has to be a thin sheet of liquid. It still has to have some windows. The transmission microscopy requires that. And so if you had more space, you would design your liquid cell a little bit differently, but the business part of it still has to be thin, even if everywhere else it could be much larger. So I think I would design the microscope with a whole lot more space around the pole piece, in which you would add all these local, essentially calibration tools, and that would be fabulous. For open cell, it's the same kind of story, but of course you have to think about can you do differential pumping well enough that you can actually do water. I showed experiments only with ionic liquids, for example, but then you need temperature, cooling, and condense the water and control it very well. So a bit like ESEM, but these things are possible, but the fundamental limitations are hard to get around. The electron beam damage, you gotta, it's not easy to avoid that. The dose limitations, and the fact that the sample still has to be thin enough to look through. Yeah. Okay.
All right. Thank you. I think we can continue then.
All right. Good. Yeah. And any other questions we can keep for the end. All right. So.
Hi.
Yeah.
Thank you for a nice talk. I'm very interested in the how to stabilize the particles in the liquid so that you can get high resolution image.
Okay, are you thinking about electrochemically formed particles that are kind of floating around and moving?
Yeah.
Okay, so
why they don't float around?
Okay, well, the question is, do you need them to float around in order for the physics of the process to be like real life? I mean, if you're doing nucleation and growth, then obviously the objects are growing on the electrode and they're kind of stuck down. But if you're doing a solution phase synthesis, let's say, intrinsically the particles, they should not be stuck down, otherwise they're not behaving the same as the same as how they are in the flask. So your only choice then is maybe to increase the viscosity of the liquid, change the temperature, maybe image at the highest possible time resolution so that you don't have like Brownian motion drift. And so you record very short exposure times, and that would require a very intense flux so that you get enough signal to, see what you need to see. So I think it depends a lot on what the kind of, what's the most important thing about the physics. If it's a surface reaction and it could be stuck down or it could be floating, then yes, you can functionalize the interior of the liquid cell to stick things down. The biology example I showed does that. Those virus particles, they were attached with a biological tether to a functionalized surface so that they could still move a little, but they weren't kind of spinning around.
Okay, thank you.
Okay, all right. Good. Okay, let me go back to the slides. Okay, and go. All right. So let's continue with part 2. So after showing you how this works and some examples, I want to talk about the sort of the hidden difficulties, not so hidden actually, all the questions that you asked, how we can address them and what's going to happen next. So the first thing I want to do is really, whenever you talk to someone, you're supposed to start with a positive thing before you give some criticism, right? So the positive thing here is that you really can get electrochemical responses from a liquid cell that look a lot like what you'd get on the benchtop. And for example, with copper sulfate, we have the correct sort of nucleation and diffusion limited signal in the CV curves. We can do these polarization curves for corrosion, and they look pretty good. And it's possible to get things that it's really important to compare with real life. And that's the basic level, is that you need to be seeing in the liquid cell what you would expect to see on the benchtop. And if not, then you have to think about, what should we do to change it. So when with our practical sort of outlook on liquid cell microscopy, I think a good way to ask this question of how valuable is this technique is to think about, why do the experiments actually fail? You're a student sitting down trying to do this stuff. What is it that goes wrong that means that you don't get good data? And my experience is that the most important difficulty, let's say, is electron beam effects. And this could include the formation of bubbles, especially when you have electrochemical stressing as well as the effect of the beam on the water. Another problem with these experiments is if the liquid layer is too unstable, like it jumps around, or it's too thick or it's too thin to have diffusion that's correct. You can have engineering challenges like poor connections, but you can also have a question about, where does the current actually flow through this complicated liquid cell? Where is the material being deposited or removed from? And so all of these lead us to wonder how it is we should go about getting higher quality and more quantitative data. And so here's what I'm going to talk about in the next section of this talk. We need to understand radiolysis. And by not just because we want to understand it, but in order to mitigate its effects, we need a strategy to do the best we can, given that we need the electrons to go through the sample to form the image. We need to understand the limits to resolution so that we can optimize that for our experiment. We can redesign the liquid cell to improve its performance. We can think about this problem that was brought up in one of the questions: what's the deal with doing electrochemistry in small volumes? And then we can increase the capabilities, like for temperature control or mixing. So these are basically the things I'm going to run through, and I'll do it fairly quickly. I think there's nothing intrinsically complex in here. It's more just to give you a sense of what people are looking at in terms of pushing for more quantitative, higher quality data. All right, so the first thing I want to talk about is this connection between large scale and small scale, which to me is one of the most important things with electrochemical liquid cell TEM. What does it mean to make these recordings, given that the volume of electrolyte is so tiny? And furthermore, what does it mean to make a recording over a square micron, like we typically do, when the whole electrode is going to be much larger than that, and there may be some hidden parts of the electrode that you can't even see? So it's important, in my view, to do some kind of a calibration. So here's an example from those movies I showed earlier. We do this for all of our experiments. Here's the current versus time transient for this movie. We can integrate the charge that flowed. We can work out how much copper it represented. We can see how much copper was on the field of view if we assume that these are hemispheres. And then we can match those things up. And amazingly enough, the electrode as a whole behaves like the tiny piece you imaged pretty closely. You get a very good match between these two things. So this is one of those sort of basic calibration things you should think about when doing these experiments. Here's another one. Here's the movie I showed of dendrite growth coming off the edge of an electrode. And we can ask ourselves, what is the local current density in the experiment? So we can count up how much copper is deposited, and we can convert that into a current density along the edge of the electrode, and we can compare that with the total current that flows in the experiment. But also we can compare the growth rates with the total current in different experiments. So if you do two experiments with two different total currents, does the material actually grow twice as fast in one case compared to the other? And the answer is yes, it does. The growth rate is proportional to the current. And so we have, again, a consistency test that helps us understand what's going on. And there's been several papers, I've only quoted one of them here actually, that discuss the issues of doing electrochemistry in small volumes. So do think about that with your experiments. Okay, so I'm going to move on from that to the electron beam effect on the experiment, which, as we discussed in the questions, is mostly an indirect effect because it changes the chemistry of the water or other electrolytes. And this could be really an overwhelming problem for us, except that there's a lot of literature out there, and the literature is there because of medical physics, where we put ionizing radiation into people, like for doing X-rays, and also for nuclear reactors, where there's a huge radiation field and there's some water coolant or liquid coolant. So there's a lot of literature to help us out. And so you look back in the papers back over the decades and understand the mechanism of what happens. The primary electrons transfer energy to water, and they transfer it in lots of little interactions, each depositing, say, 20 eV worth of energy. Remembering that the electrons started out with 200 kV energy, they do not stop typically in the sample. They pass through, they lose a tiny bit of energy, they lose energy along their path. And so this makes it quite easy to calculate how much energy is being deposited into the sample. And it's a lot of energy. So here's the dose rate for a typical TEM experiment. The gray unit is the energy absorbed per mass. And this is the electron flux density. This is typically used when recording, say, a movie. Okay, so this is, so this number, 10 to the 7. Let's compare 10 to the 7 grays per second to what we would see in medical physics. This is the grays per second you might get from taking an X-ray of, like at the dentist's office. And this is the dose rate you get in the walls of a nuclear reactor. So just think about that for a moment. The sample is in a radiation environment that is 10,000 times stronger than being in the wall of a nuclear reactor. So kind of awesome that anything survives at all. So moving on from there, the fact though is that only a tiny volume is being irradiated. So the total energy isn't high, it's just the energy density and rate that's high. Okay, so what happens to this energy? It ionizes individual water molecules, it ejects electrons from them. These ionized molecules and ejected electrons transform the nearby molecules into radical and molecular species. And these are the initial yield species. This is the hydrated or solvated electron. And along the track of the electrons, they react with themselves and with more water to get more species. These all diffuse around and get uniformly distributed locally. And then after longer time, you get secondary reactions to form things like oxygen. So this is very well known for water. Notice that there's nothing apart from water in this. I'm not talking about electrolyte, I'm just saying plain water for now. How do we model this? Well, we have all of these species that I listed, plus a few others. They all react with each other. They can be destroyed by reacting with others. They can be formed through reactions of other species. And of course, they're being formed by the radiolysis reaction by the electrons, and they can diffuse away. And so this allows us to calculate the concentration of every radiolysis species using convenient tools out there. And so these calculations, which are based on empirical reaction rates and such like from the literature, tell us a lot about what's going on. So what if we illuminate the whole volume of liquid with the electrons, for example, in the little pocket in a graphene liquid cell? This is what happens typically. Look at the time range. All of these species started out with very low concentration. After some time, they build up to a certain concentration, depends on the dose rate, but they build up over less than a millisecond. So it's almost instant that the electrolyte goes to a steady state with a different chemical composition. And the reason it's a steady state is because all of these things are in steady state. They're being destroyed and formed, and this happens until there's a balance, until the concentration of everything is correct to keep this in steady state. So a steady state forms. Given the speed at which things form, it's clear that liquid flow can't remove radiolysis products unless you could flow at 1 meter a second. The other thing is that this steady state concentrations depend on the dose rate. And even, so we used 10 to the 7 before, even if you reduce the dose rate really low, it's still going to be quite an effect. You cannot image with so few electrons that you don't change the chemistry. Okay, so this is sounding a little bit grim, right? It goes bad immediately, and however low the dose of electrons, it's not low enough to make things better. And these species contribute to different things. Obviously, the hydrogen could form bubbles. The hydrated electron could reduce ions, and the H+ that's created could change the pH. If we now do a calculation where we only illuminate a little bit of the sample, like in a STEM or in a TEM with a small probe, you get similar results within the probe and some kind of transients around the edges. And so to summarize, there are lots of consequences of the electron beam striking the electrolyte. And this is one of the most interesting ones to me, the changes in pH of water as a function of the dose rate. So for example, supposing you start with a solution that's acidic, the water doesn't really, the illumination doesn't really change it. But if you start with something that's alkaline and you go to the dose rates that we use in the microscope, something that started out at pH 14 is going to end up at pH 10 or 11. So dramatic changes that could happen during, just at the moment your beam goes on the sample. Okay, so here's an example of one of these experiments. This is the reduction of metal ions into particles. And what we're doing is we are applying a voltage to an electrode, which is up the top here. We are actually, excuse me, using the voltage to release gold ions into solution. And then the ions diffuse away, they've diffused to here. And now the electron beam is illuminating the electrolytes, and the hydrated electrons are reducing them into gold particles. And we can turn on the voltage. We're actually turning on the source of the ions by switching the potential off and on, the current off and on. And you can see that the growth is they only grow when the voltage is on. So it's kind of an interesting illustration of the process of releasing the ions into solution at a certain time, and then they diffuse along, and then the electron beam reduces them back into particles. So you can, in principle, maybe track the diffusion of the ions off the electrons, off the electrode. Okay, so I want to show you this slide because it's possible to do some interesting calculations on radiolysis. And one of the ones that we found interesting was how many of these radiolysis products are there in your experiment. So specifically, let's set up an experiment where the electron beam illuminates an area. The area contains gold ions. We're using gold chloride solution. The beam creates hydrated electrons and they reduce the gold, and it forms a single island. And it's very easy to measure the volume of the island. And if we do all the calculations, we can find that it takes about 6,000 primary electrons to reduce each gold ion into a gold atom. And there's of the order of 50 hydrated electrons present at any time doing this job. They're created and destroyed very, very quickly, but this is the equilibrium number. Okay, so that's kind of a fun thing, and it's a little bit off the point, but it's just to show that you can get some understanding of what's going on, and it's all consistent with the radiolysis calculations. So how can we mitigate beam effects? Well, there's no single solution, and so depending on your experiment, you should choose a good strategy. For example, you could use low dose imaging, which means don't keep the beam on, just take a quick picture and then move it away or turn it off. You have to compare with unexposed areas so that you know that what you see is real. You should always record the images with the minimum signal to noise that you need to show what you want to show. So beautiful images tend to require higher dose. And so when you see noisy images, you know the person is doing the job correctly. You can use chemical scavengers to reduce the effects of the radiolysis products. And you can, if you have a choice, choose electrolytes that are less sensitive to the beam. Of course, count every electron, use direct detection camera, but those are expensive. But if you can find a microscope that has one, you're in better shape. And then we need to do a lot more modeling for less familiar electrolytes, including if you add a whole load of ions into the solution, how do they change radiolysis? What happens with ionic liquids? So there's a lot of work going on here. I've just cited a few of the papers that I've found useful over the years, but this is a very active area, and people really, every experiment you've got to think about the beam effects. But so I've already referred to the fact that the sample you know, the microscope is going to be changing the sample as you do the imaging. And so that makes us think about what is it that really limits the resolution of the images in a liquid cell experiment. And for very thin samples, or for samples where you don't care how many electrons you use to make the image, the resolution is then limited by the performance of the microscope. But it's much more typical that the resolution is dose limited. In other words, you cannot shine enough electrons on it to see your object over the noise, because the sample is too sensitive to the electron beam to be worth imaging after you've illuminated it with so many electrons. And so we typically, like the biologists, have to think about dose limited resolution. And that leads to graphs like this, where you say, what is the damage threshold for certain types of materials, and what kind of image resolution could you get if you can only use this many electrons to form the image. And so you often find that understanding what limits your resolution is a very important aspect of the experiment, because if it is dose limited, you really have to use low dose techniques. And if it's not dose limited, you can then go and use a higher dose and get a nicer looking image that would have a better resolution. So having said all of that, you can actually, this is kind of a version of that graph I showed earlier, but it's a lot uglier and more complicated. But this type of calculation allows you to say, should I use TEM or STEM? What kind of imaging mode should I use? What kind of thickness should I use for my sample? What resolution do I expect to get? And these graphs do show that, for example, for the right kind of objects, you can really see atomic resolution. But for many typical objects, especially biological ones, your resolution is never going to be that great. It's going to be a few nanometers. Okay, so having said that, if you get the chance to do liquid cell microscopy, you still want to pick the best microscope you can. Definitely do your experiment in a modern aberration-corrected microscope, because aberration correction, although you tend to think of it as a way to improve the, let's say, the ultimate performance of the microscope, it also is a way to improve the interpretability of the images. And if I compare these two images, you can see that they both show where the atoms are in this random sample. But look at these edge effects in an uncorrected microscope, conventional imaging. The only way to really work out where the sample ends is to do a simulation. If you have CS corrected microscope, you get much easier images to interpret. And this is true, this is a vacuum experiment, but this is the same thing in liquid. This kind of fuzziness around the edge of the particles is caused by the aberrations. And if you can correct the aberrations, you get much more easier images to interpret. If you can do chromatic aberration correction, you're definitely in a good place, because this helps a lot in terms of using all the electrons that maybe lost a little bit of energy in the sample. They would not be focused into a good image in a normal microscope, but a microscope with a chromatic aberration correction allows you to use all of the electrons, even the ones that lost some energy. So the only trouble is there's not many microscopes like this out there. And they're very hard to get onto, and maybe the users don't want you putting a liquid cell in there in case it breaks. So there are other ways around. You can do energy filtering, but if you do have access to this kind of microscope, that would be a good one to use. Okay, so I talked about the resolution and I talked about the electron beam effects, but for the next part, I want to talk about a little bit about the hardware. Because after all, the design of the liquid cell that I showed you earlier is the same design as from the 1940s, 2 windows with a spacer layer that enclose liquid. So how can we improve this? It's been clear from our discussion of resolution that if you want good images, you want to have great control of the window thickness and the liquid thickness, as well as all the microscope parameters and the electron beam effects. But let's see what we can do in terms of engineering the window and liquid thickness. So one way, of course, is to use 2D materials for windows. So here's graphene stuck on both sides of a silicon nitride spacer to make very small enclosures for reactions. This type of experiment so far hasn't been that good for electrochemistry, because we really want to be able to flow our liquids, heat them up, put an electrode in there, have a full circuit, and that's more difficult with these complicated liquid cells. But another way of controlling the liquid thickness is by changing the pressure, because we are pushing the liquid in with a syringe pump. And it turns out that if you pump also on the exit as well as the entrance, you can have a much better control over the liquid thickness. And I think, this is kind of happening recently. Again, these are just some citations. I hope I don't offend somebody by missing their paper. I know there's other work on this, but here are 3 papers that describe approaches to controlling the liquid thickness. I think this will have a transformative impact on future liquid cell microscopy, just the ability to control where the liquid is and how thick it is. So here's an example of what you might call a typical complicated, hard to interpret experiment. During the electrochemical process, which here is growth of gold particles, you can see that the liquid is jumping around all over the place. And this is generally bad, because we like to know where our liquid is for the electrochemical reaction. But as someone pointed out in the chat, you can use the motion of the liquid as a way of controlling your experiment. You could, for example, have a fully filled liquid cell. You could do an experiment, and then you could generate a bubble that would displace the liquid temporarily, allow you to record some high quality images, and then go back to a thick liquid layer. So controllable formation of bubbles is a promising way to control liquid geometry, but taking care of wettability is also essential. We also could look at improving our electrode. Sometimes you don't have a choice, but sometimes you could change the electrode material. And obviously, the thinner it is, the better contrast the growing objects will have, the less dose you'll need to image them. Everything will be much better. So I'm showing you now a movie of copper growing on a graphene electrode. Notice a few things about the movie. It's very noisy because we're using low dose. It's got very low contrast for the electrode, which ends here. Let's play it again. I like this one. And you can see so many details in these particles that were not visible in some of those movies I used earlier. So graphene is a good electrode for liquid cell microscopy. You just have to kind of transfer them by hand, but you can use them. And here's some examples of the improvement in resolution that you can get from using graphene electrodes. So now I'm going to talk about temperature. We mentioned this briefly in the chat. I said that for us, a high temperature is like 100 degrees C. Liquid cells have been used for heating for quite early on, but people have typically not done heating plus electrochemical experiments. And one of the reasons is that the heater circuit needs 2 electrical connections. The electrochemical experiment needs 3 or 4 electrical connections. You just don't have enough of them in typical liquid cells, but these things are changing. And here's some examples of what you could do with heating capabilities. So the heating is done using a platinum heater that is aligned with the window and has little holes in it. So ordinarily you would be looking, you don't want to be looking through a heater made of platinum because that's a lot of material to look through. So you have the heater circuit cut with tiny holes, or you have it maybe on one side of window so it doesn't block the view very much. And then you add the electrodes on top, and there's a silicon nitride layer to keep everything insulated. So that's how you might include heating along with electrochemistry. And here's a slide showing that reaction, the etching reaction of nickel that I showed you a movie of earlier. And this being done at 2 different temperatures, you can see a somewhat of a difference in the kinetics of the etching of individual objects because of the higher temperature. Here's another example where we deposit copper on one of these heated windows, and we just measure how fast it deposits. And what we find indeed is that deposition is faster if you heat up the experiment. So not really surprising, but at least it's a proof of principle experiment. In this case, the heated region is much smaller than the total electrode. And so this may be a way of kind of favoring reactions only on the parts of the experiment that you can actually see in the microscope. We need to know what the temperature actually is. And this is typically being addressed with finite element calculations. And so here's the heated distribution on one of these little platinum strips. And here's the heat distribution vertically in the sample. So the heater is only on one side. And you can see that there's actually a gradient in heat from the hot to the cooler part as you go vertically. So this would favor reactions maybe on the electrode that was close to the heater. Okay. And of course, temperature changes everything. One thing it changes is radiolysis. Most radiolysis species actually become less concentrated because the reaction rates are faster. And so this tends to mean they get consumed faster and therefore have a lower steady state concentration. So I'm not saying this is a good way of reducing the effects of the electron beam on the electrolyte, but it is a way, it is interesting to consider that the radiolysis effects are lower at high temperature. Okay, so I've talked about some engineering improvements in the liquid cell, but I want to now talk about a bit more kind of larger picture view of what else we can do with liquid cell microscopy, just to expand the capabilities and give more opportunities for quantitative and accurate data. So one thing that we've come across, we've discussed in the question session as well as a little bit earlier, is combine your liquid cell with other techniques. You know, there's so many different techniques to use out there. One fun thing is, can you take your liquid cell tip with the exact same experiment and move it to another instrument? And here's an example of how a TEM holder has been placed in an X-ray microscope to compare the lithiation of these particles that, sorry, I've forgotten exactly what they were to start with. But correlation is a really powerful way of combining the information from electrons with X-rays, scanning probe techniques, optical microscopy, and other kinds of stimuli or measurement techniques. Go beyond imaging. This is the same compositional mapping example that I showed earlier, where you can try and look at the chemistry changes during battery operations. Here's an example using energy dispersive X-ray mapping, and you can see the difference in composition of this object. But there's a lot of electron beam effects in at least in this experiment, because a very high dose is required for compositional analysis. We can rethink the sample completely. People are really good at microfluidics, so why not use microfluidics in your liquid cell so that you can pump certain volumes of liquid to certain places and then image what happens when they react. This could control liquid pressure and the geometry and the amount. You can do titration experiments, very sophisticated. You can make your experiment into an electrochemical experiment in many other ways. And I want to show this one because it's very close to my heart. This was one of the things that got us interested in liquid cell microscopy, we were very, let's say we were enchanted by the morphologies you can get when you do anodic etching of silicon. You get these you get these channels, you get side branches, you get these dendritic structures, and the details of the structure determine the properties of the material. So how can we look at anodic etching in situ? Well, we could design a sample where silicon is only etched in a certain region because of the doping, and then we can use that silicon integrated object as the sample. And so the experiments didn't really work, but this was one of the things that got us thinking about assembling a liquid cell to put the electrolyte in. But really, there's no reason not to do this type of experiment using other materials where they kind of form their own encapsulation. And then finally, cooling. We talked about heating. You can do cooling of liquids using small Peltier coolers, and you can thereby replicate some of the capabilities of environmental SEM in terms of condensing water. And this, I think, is very important for things like corrosion processes, which do take place in a water-air environment, and you want to condense the water out of the humid air. So let's move for the last few slides onto some really speculative things that I think could just be fun to think about, even if we never ever do them. As you know, electrochemical deposition can change the stress state of materials. Microscopy is very sensitive to small changes in stress or orientation. And so it's possible with a cantilever electrode to measure stress as you do a deposition experiment. It's possible to measure the chemistry of the electrolyte because the transmitted intensity is sensitive to the concentration of especially heavy ions in solution. And so you can see some effects of depletion of electrolyte in real time during the movies. And all of this leads me to think that there are so many grand challenges and little challenges that we could do with liquid cell microscopy that people so far haven't really pushed on very much. When I was talking to people over the years, I sort of got intrigued by things like Janus particles, at electrolysis processes that you could trigger by changing the temperature, at co-deposition. But the most fundamental one, the electrochemical double layer, truly a grand challenge in electrochemistry to image this. And so you need an atomically flat surface. You need the electrons to go parallel to the surface. You need extreme sensitivity, and you need to account for the beam effects. But I think it could be done. And I know some people are going along this pathway, and I think this will be transformational. All right, so I'm going to conclude there. I think that the points I've been trying to make, I hope I've transmitted that. I think the opportunities and challenges both are extreme, right? There's some fabulous things you can do with this technique. There's some severe challenges that you have to account for in interpreting your results. But for all of the challenges, we have some kind of a workaround, or at least a mitigation strategy. And this leads to some very exciting properties. So we can address optimization of resolution using the instrumental advances in microscopy. We can sort of address the beam effects through understanding and simulations, even if we can't eliminate them. And we can now control our liquid environment much better than before. We can apply temperature, we can control flow conditions, and we can use other strategies with respect to designing the cell to improve the control. So I think the prospects are really exciting, and hopefully this will stimulate more people to get involved with this. It is a long pathway, but I feel like the results are worth the effort. All right, so I'll stop sharing now, and I'll be happy to take other questions.
Thank you so much, Francis. So we're open for more questions. I think I don't know how much time we have, but Francis, you should tell us if we're going too far.
Oh, no, that's fine. That's fine.
That's okay. Yeah, maybe I can start by asking a very typical question that I think is still always is on our mind. So there are two ways that I would say that you can do electrochemical experiments. One is when you just apply bias, you run the current and you look at what you're getting, but your focus is what you're getting. So you're, for example, diffusion, the growth, corrosion, and so on. And the second is when you also reference your potential to a specific thermodynamic value, and then you actually get the actual electrochemical data. So electrochemistry then comes as an additional data set that you can use to interpret the data, basically. So in your impression, how close we are to getting the right potential reference inside? Because quasi-references are not quite references. Using 2-electrode cell is just to see the reaction, that's fine. But what if we want to really correlate the electrochemistry? So the potentials, the thermodynamic state of the system.
Right. Yeah, actually, this is really a fundamental question, and probably I should have emphasized that a bit more. At the most basic level, it's always good to see some kind of electrochemical measurement along with the movies, because otherwise you really don't know where the current is going and where the voltage is being dropped in the system. But even if you record current versus time or a CV curve, you have a question of what is the true reference? Can you do anything with the geometry of the liquid cell that would give you a real reference value? And so there's a constraint in terms of the materials and the space available within the local part of the liquid cell where you're doing the imaging. And that's why people tend to rely on these quasi-reference electrodes. And typically, at least they could be built out of a material that would be reasonable, at least won't have a whole lot of electrochemical reactions taking place. So it gives you some kind of essentially a calibration. But what's really needed is a proper reference electrode, which actually has some volume to it. And the closest place you can put it is near the tip of the holder, but not right there. So all those holders, they get a little bit wider. In some designs, we've used one by Hummingbird. There's a small reference electrode just where the sample holder gets a bit thicker, and so the liquid stream goes past it. So it's not perfect, but one thing that we have been able to do is in a well-characterized system, you can measure what you get on your quasi-reference electrode at the same time as this real reference electrode and see, and that gives you at least some sense. So I think that your question is a very good one, and it's not an easy thing to solve, but the best we can do is probably a close by but correctly designed reference electrode.
So there is some way to go, basically.
There is some way to go. Yeah. Yes. Yeah.
Okay. There's a question from Amir.
Thank you, Francis. Again, about the electrochemical reaction, is it possible we monitor electrochemical reaction from the, I don't know, it's a biomolecule or molecule, we monitor them? I think it's not possible, huh? To, for example, enzyme activity or something.
Right. I think that these kinds of biological electrochemical things are a lot more difficult because the materials, the changes that take place are going to be more subtle. The materials typically have very low contrast, and even if you were just trying to do a biological imaging of that thing, you would use cryomicroscopy, and you would typically have to record images of many different things and do some averaging and use some of the techniques that the biologists have developed. So it's definitely a challenge. I don't know if I want to speculate whether we'll solve that challenge in the future, but it has been remarkable what the distance that we've come so far in looking at things that we didn't think would be possible. So I feel like learning some tricks from the biologists would, applying those within this situation. The biologists freeze the sample in order to immobilize things, and also in order to reduce radiolysis effects. And then they use very low dose imaging. So whether you can do, for example, freezing to kind of fix something in place and then image it at that point, I don't know if that's a feasible solution, or whether I've seen experiments recently where a laser beam locally heats up just one small part of a frozen sample to get that part going, and maybe something along those lines would work. So if we were really thinking long term, perhaps there's some kinds of new stimuli that we could use to do that kind of imaging. But you're asking, I think, a really hard question given the current state of the knowledge.
Because I'm working in intracellular electrochemical measurement, but I know it's a routine. My experiment is very hard, but when I imagine if you want to combine it with TNO, it will be very big challenge. But it's maybe one day.
I think so. Yeah.
Thank you, Francis.
All right. Thanks.
Jan, please go ahead.
Yes, thank you. Great talk, Francis. I wanted to ask a question about the graphene cells. You showed some examples there. Do you think we'll ever be able to use the graphene, the multilayer graphene, as a window and as an electrode at the same time? And what do you think are the challenges there to solve?
Right. That's a good question. I think that it's better to separate the functions, and the reason is that there's often a benefit to having a small area of the electrode, because if you consider the electrode as kind of using up the species in solution, you run into difficulties with diffusion more quickly if you have a large electrode. So maybe the current that flows in the circuit is larger, and the number of ions that get discharged is larger, and then you run out of ions in the electrolyte. So people often look for ways to reduce the kind of accessible area of the electrode, and therefore the window typically is a lot larger. You want to be able to look at places on the electrode, away from the electrode, and you have to you also have to design your window geometry to accommodate, for example, the limitations of the etching of the silicon, which means that you can't control really small windows. So I feel like there's engineering issues that are different for both, and it's probably easier to separate the effects. Graphene's not the only one that is used as a window material. Many of the other 2D materials have, similar mechanical properties, but many of them are insulating. So you can imagine a composite of 2D materials would be a good way to do it. Graphene on HBN or something like that could be a good way to go, actually.
Okay, thank you.
That's an interesting question. Yeah. Actually, while I still have people, there's going to be a lot of discussion of this kind of thing at a Gordon conference in January. Let me put in a quick plug for that. And it's like a 5-day meeting. Here it is. Don't know if you see that. Yeah, good. All right, here's the dates of it, end of January. So this is in a beautiful part of California. And so it will certainly have, it's not only electrochemistry, but it will cover all aspects of electron microscopy of liquids. So a little bit of an ad for that. Okay.
Yeah, great, thanks.
Yeah, good. Any other questions?
Yeah, let's see. Maybe I can ask one of the last questions, then we'll see.
Sure.
We have to wrap it up. So if I try to look at the in situ microscopy from the sort of less exciting side. So it's a lot of trouble, it's a lot of effort. And at the end, we get basically movies, right? So ultimately, it's the movies. We're not getting like crystallography, crystallographic information, or chemical information is really hard to get. And so would it be maybe more feasible to go with ex situ microscopy, even though it may be identical locations microscopy, but you can, yes, it takes a bit more time, but at the same time, it's much less efforts, much less struggles.
Right.
What would you say then?
Yeah, I like this question, because every in situ microscopy project, you have to think about that. Is it really worth all this trouble that I'm going to? And I would argue that there are several ways where, excuse me, a movie is much more than the sum of just a series of stills recorded in gaps between reactions. And one of those ways is the actual kinetics, the measurement of how fast something happens. Assuming that, so let's say, what is the rate at which this facet is growing compared to this one, or the the change in volume or dimensions of something. Those things are much harder to get if you do a little reaction, you stop, take a picture, do a bit more, because you have all this change in the conditions. So kinetic information is much harder from stills. And the other thing is that many configurations only persist during the reaction, and we see that really clearly in let's say crystal growth experiments in vacuum. And so I think the same kind of thing applies for electrochemistry. If you do a deposition experiment where, for example, you're doing chemical vapor deposition in the microscope and you have a movie of something growing, if you cool it down and turn off the flux, the structure changes and the interrupted image is not the same. So I think that there's a real benefit to having this continuous view of the sample, if you can, to make kind of detailed physical measurements. And the other thing, I guess the third thing that you find when you're playing on the microscope is that you can, if you're seeing it happen, you can tweak some condition. The growth isn't going as you wish, you just change something. In that sense, as a combinatorial experiment where you're doing many different growth experiments all at once, you can indeed save a little time compared to a like before and after shot of each one of those things.
Okay.
So, but definitely,
agree.
Honestly, for any scientific thing, it's worth saying, am I doing this because this is what I always do, or am I doing this because this is actually the best way to find out what I need to find out?
Okay, and Francis, would you say that it's possible, because I've never done this kind of experience, I'm just curious, is it possible to add a micro bubble into the line with electrolyte? Maybe it's somehow, let's suppose we could do it and just flush it and get to the point when you remove the electrolyte, do the imaging, move it further, get the electrolyte again. And so of course your electrochemistry should be working. That's a different story. Let's suppose you just do a liquid experiment with crystal growth. Would it be still feasible, or the windows will break quickly?
I think it is feasible. It is feasible, and there are people doing this kind of thing. And you want to generate it locally, and you preferably want to be sure you can get rid of it afterwards. So one possible way is to generate it electrochemically. So you might even have an electrode in there for the purpose of making doing evolving hydrogen or oxygen in your electrolyte. Just for a moment, make a small bubble, push things around. You can sometimes, if there's a suitable nucleation site, use the electron beam to trigger a bubble. Or you could, you can inject gas and liquid through two different portals into the system. But the problem with generating these things externally is you don't really know how it's going to flow into the viewing area. It's quite far away. But I think that the control of the environment through different pumping schemes and injection schemes, and maybe even like a little hot wire that you can use to boil stuff locally, all of these methods allow for a little bit more of a design of the liquid environment. Like you want it here, and then you want it to be gone to record an image, and then you want it to come back again and do a bit more. These things are possible.
There was a really nice question from the chat. So it's asked if you can, so you're getting 2D images. It's like projections, basically, right, of the entire cell. Can you reconstruct to some extent the 3D shape of the particles or whatever you grow, the crystals?
Yes.
Which is completely inaccessible with contrast issues.
So there are two. So you can do tomography, and it's very well set up in the TEM that you would get a series of tilts, and then you would integrate the information from each one. Now you have an issue with a liquid cell in that the geometry of the liquid cell, as you tilt, it gets much thicker, and so the image quality gets worse. Whereas a normal tomography sample is kind of like a tube, and you rotate it and it's still the same thickness. So that's an engineering problem, and there are indeed some efforts there to make a liquid cell where the liquid isn't in a sheet but in a tube. That can be done. But perhaps the more important limitation is just that every one of these images requires another dose of electrons. And so there would be certainly some samples that were not dose sensitive, but for many applications, it's the same problem as with compositional mapping. It's just that you have to use a lot of electrons to achieve the measurement, and then that can cause its own trouble. Yeah.
All right. So maybe one more last question that we have. So Ismail, go ahead.
Yeah, thanks. I have a question. Just what would be the challenges if you want to bring the kind of optic fiber just next to the working electrode? I mean, in terms of microscopy and imaging. I mean, I'm just talking about like photoelectrochemistry, basically. If you want to just do the pulse, the laser pulse, basically.
Right. Thanks. So I know that there are some microscopes. You know that ultra-fast microscopy is often done by having a laser that hits the filament and give a burst of electrons, so that the laser is also able to pulse the sample so that you can do pump probe experiments. So there are microscopes out there where the sample is accessible with a laser beam. So it's not really even an optic fiber. You just have to build it like a direct line of sight to the sample. So that can be done. And these can be reasonably powerful lasers. I think you have to worry about, are you putting a lot of heat in there at the same time with the windows and the electrolyte and the does the sample get hot and start to drift, or hot and start to boil or something? But assuming that's under control, there's no intrinsic reason why you can't get a light excitation onto the window of a liquid cell. And so there's a couple of papers, I think, where this kind of thing has been demonstrated. Yeah.
Thank you.
I think that's exciting. I always wonder with photons, the photons would have some effect on the sample. The challenge is making sure that the electrons that image it don't have, if it's sensitive to being lit up by photons, I would think the electrons would probably have a pretty strong effect on it too.
Yeah. Thanks. Thank you.
Okay, thank you so much, Francis, for this very exciting colloquium that you led. It was really nice. I think with this, then we're going to wrap it up. Thank you so much.
All right. Well, thanks for the opportunity and the really interesting discussion. I've enjoyed it and appreciate it.
Very good talk. Yes, we really enjoyed it. It was great to see so many questions. Thank